everyone. My name is Soumili, and I'm a co-op student at the WU Lab at Dana-Farber Cancer Institute, and I'll be discussing my CaNCURE project on investigating clonal dynamics of resistance in heterogeneous ovarian cancer cells using a multifunctional lineage tracing tool. Ovarian cancer is the 7th most common cancer and 5th deadliest cancer in women. The current standard of care that's being used is platinum-based carboplatin, which is a DNA cross-linking agent, and paclitaxel, which induces mitotic arrest in cells. These two chemotherapy drugs are used in combination with surgery to treat ovarian cancer patients. Response rates to chemotherapy is quite high, which is around 80%, but the survival rate in patients is still quite low, around 30%, as you can see here in this graph. And a part of the reason for this is due to the heterogeneity of tumors. As a parent cell metastasizes into a tumor, they may acquire mutations along the way that gives it acquired resistance that allows it to survive several rounds of chemotherapy. Similarly, some clones might have pre-existing resistance, and this also allows it to be selected for and survive several rounds of chemotherapy. This leads to our question that's being asked about what are the evolutionary trajectories in the clones of ovarian cancer cell lines when they're treated with platinum-based chemotherapy, and what do these trajectories reveal about resistance mechanisms among these clones? So this is an overview of the experimental workflow that was used. 2 ovarian cancer cell lines were used, namely P1 and JOS2. Cells from these 2 cell lines were transduced with DNA barcodes, and upon transduction, they were subject to 2 rounds of chemotherapy. Cells were collected prior to any chemotherapy treatment, that's called T0, and post 2 rounds of chemotherapy called T2. Next, one of these clones from the JOS2 cell line were chosen for drug sensitivity experiments. In the first graph here, you can see that the cells were treated with carboplatin and paclitaxel in combination, and the T0 clones performed more poorly than the T2 clones. A fewer number of cells survived over the course of 9 days of treatment. Similarly, in these clones, when they were only treated with carboplatin, the T2 clones survived better than the T0 clones. However, with the paclitaxel treatment, all of the clones performed similarly, which can lead to the conclusion that carboplatin is driving the selection of the T2 clones. And this is a potential resistance mechanism that the T2 clones might have taken. So they might have acquired resistance post second round of chemotherapy or acquired resistance after the first round of chemotherapy that allowed these T2 clones to survive both the rounds of chemotherapy and still proliferate. So the key takeaways from this experiment and many other experiments that were done through this project was that clonal evolution leads to development of resistance that causes selection of clones to survive chemotherapy. And these resistance mechanisms that can emerge among these clones varies. In the example shown with tofu, the resistance was acquired, but there were several other clones tested that had pre-existing resistance that allowed it to survive several rounds of chemotherapy. So understanding these chemoresistance pathways can lead to development of more robust and effective therapies for ovarian cancer in the future. And through this co-op, I had multiple personal takeaways where I was able to learn various technical skills, namely cell culture, chemotherapy treatment, CTG assays, etc. I also have a greater understanding of clonal evolution and how that might look like during metastasis and other mechanisms that contributes to therapeutic resistance in ovarian cancer. I also have an increased awareness of the pharmaceutical landscape, cancer nanomedicine, and applications of nanomedicine in other fields. And from this co-op experience, I developed a research interest in cancer pharmacology that I hope to pursue in graduate studies in the future in my career. So I would like to thank the CaNCURE program and everyone in the WU Lab that helped me support this co-op research experience, and I'm very grateful for this opportunity. Thank you.